If I say CRISPR, what do you think of? Maybe it's designer babies, curing disease, rewriting genomes, or that one movie with The Rock. But that's not what CRISPR really is. CRISPR didn't evolve for the purpose of editing genomes. It evolved because microbes everywhere are under constant attack. For billions of years, bacteria and archaea have been locked in a microscopic war with viruses. A war so intense that it prompted the evolution of one of the most sophisticated defense systems known to biology, CRISPR-Cas. Viruses are everywhere, and they infect more than just humans. To give you an idea of just how abundant viruses are, it's estimated that there are around 10 to the power of 31 viruses on the planet. That's more viruses than all other organisms combined. Wherever you can find microbes, whether it's in the oceans, the soils, or in the human gut, there are viruses there infecting those microbes. And predation by viruses can be both fast and lethal. You see, once a virus recognizes its host cell and attaches to it, it injects its genetic material into that cell, and it hijacks the machinery within to produce more copies of itself. It can do this up to dozens or hundreds of times before it bursts the host cell and releases all of those new viruses back into the environment. From the perspective of the microbes that are targeted by these viruses, this is a potential extinction level threat that they need to address, and it's happening constantly. As a result of this, they've evolved a myriad of different defense systems to protect themselves from viral predation. Aside from CRISPR, there are of course other defense systems that microbes use. For example, microbes block entry of viruses into their cells by modifying the way that their cell surface looks, effectively making a disguise so viruses can't recognize them. They can also use things called restriction enzymes, which recognize when DNA or RNA inside the cell is from outside the cell and destroy it. Or they can adopt things like abortive infection processes, where they recognize that they have been infected by a virus, and the cell that is infected chooses to kill itself in order to protect other members of its species around it. These defenses work sometimes, but they can be slow to adapt, and viruses are amongst the fastest evolving organisms on the planet. CRISPR provides a solution to that problem by turning previous infections into future protection. But what exactly is CRISPR? Well, the name CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. You see, when researchers first started sequencing microbial genomes, they noticed these regions of the genomes that stood out because they contained the same short DNA sequence repeated in multiple locations, spaced out by unique sequences. These unique sequences, because they spaced out the repeats, are referred to as spacers. Despite noticing them early, it took around 15 years for researchers to really understand just what these arrays were. But eventually what they noticed was that these spacer regions actually corresponded with 100% identity to the genomes of viruses that the cells that carried them had previously been infected by. The CRISPR arrays are, in fact, an archive of previous infections. Next to CRISPR arrays are proteins that are called Cas proteins, CRISPR-associated proteins. If the CRISPR array is the immune system's memory, then the Cas proteins are the mechanism for turning that memory into immunity. But how exactly do these CRISPR arrays actually work? Well, when a microbe survives a viral infection, something remarkable can happen. Fragments of the viral genome can be captured by Cas proteins and integrated into the array for future use. The proteins responsible for this are Cas1 and Cas2. Cas1 is a nuclease, while Cas2 can be either a single-stranded RNA or double-stranded DNA endonuclease. In the type 1e system of E. coli, Cas1 and Cas2 form a complex where two Cas1 dimers are linked with one Cas2 dimer. The Cas2 dimer binds to double-stranded DNA, and then the Cas1 dimers bind to single-stranded regions on the flanking sides of that DNA and help to integrate it into the CRISPR array. Once it's integrated, that spacer is now part of the cell's genome, and that means that when the cell divides, it's inherited by the daughter cells. This means that future generations benefit from the immunity that the cell has now obtained. This isn't immunity like we know of it in humans. There's no antibodies, no immune cells, but it is an adaptive immune system. And it's actually one of the clearest examples we have of Lamarckian-style inheritance in modern biology, where experience shapes heredity. Now, having a defense system is no use if it's not armed and ready to go when you need it most. And to arm a CRISPR array, the first thing that has to happen is that the spacers themselves have to be processed into mature CRISPR RNAs. The first step in processing them is for the entire CRISPR array to be transcribed into RNA in one go. After this, the individual spacers are then processed out of this CRISPR array RNA. The way that they're processed varies depending on the type of CRISPR system we're talking about. In some systems, flanking regions to the spacers form three-dimensional structures that we refer to as stem loops, which are then recognized by proteins Cas6e and Cas6f. They target a specific site for cleavage in order to produce the mature RNA. In other systems, the Cas6 proteins are still involved, but instead of stem loop structures forming, the RNA actually wraps around the Cas6 protein and targets a specific site for cleavage that way. In type 2 systems, they have a completely different process, where an additional RNA, called a tracer RNA, binds to a region next to the spacer, and by virtue of where it binds, targets a site for cleavage. Whichever way the CRISPR RNAs are processed, they then form part of complexes composed of them and Cas proteins that recognize foreign DNA and are ready to act as surveillance complexes. Think of them as molecular patrol units, each of which carries a unique viral sequence that it recognizes and is ready to destroy if it comes across it again. At this stage, nothing has happened, but the system is armed and ready to go if a virus does infect the cell. Now, as I said earlier, when a virus infects a cell, it injects its genetic material, DNA or RNA, into the cytoplasm of that cell. And if one of our surveillance complexes from the CRISPR system recognizes a sequence in that virus's genetic material, then it can activate. The way that this occurs varies depending on the type of CRISPR system that we're talking about. For example, in type 1 systems, an additional sequence called a PAM is required in addition to the target sequence. If the spacer anneals to the target sequence and the PAM is also recognized on the opposite strand to the target sequence, then a cascade is formed where the protein complex changes shape and Cas3 is recruited in order to degrade the DNA. In type 2 systems, a single protein is used in order to carry out the degradation of the DNA, Cas9. In addition to this, like type 1 systems, Cas2 systems also require a PAM, but in this case they require it to be on the same strand as the target sequence, not on the opposite strand. Again, if both the PAM is present and the target is present, then Cas9 is recruited and DNA is degraded. Type 3 systems, like the type 1 systems, require multiple proteins to carry out their activity. But, unlike the previous two systems, they seem to target mostly mRNA, not DNA, although they have been shown in some experiments to target DNA as well. The exact mechanism of action for the type 3 systems is something still being explored. And so, different CRISPR systems accomplish their goal in different ways. Cas9 cuts double-stranded DNA. Cas12 creates staggered cuts in the DNA. And Cas13 targets RNA instead of DNA. And so when we talk about CRISPR, we're not talking about a single thing. We're talking about a whole family of different systems that exist for different purposes. There's an obvious danger here. CRISPR systems target viral DNA. So why don't they target the cell's own genomes that carry the spacers within them? The answer to this lies in those little motifs that I referred to just before, the PAMs, protospacer adjacent motifs. See, CRISPR systems don't just randomly acquire viral DNA from anywhere in order to incorporate as spacers. In order to incorporate them, they need to have a PAM adjacent to them. PAMs are short regions of 3 to 5 nucleotides that are unique to each CRISPR system. Protospacers are excised from adjacent to these PAMs using a kind of ruler approach, so that only the spacer is included and not the PAM itself, and the correct length of spacer is excised for being incorporated into the genome and to the CRISPR array. So what that means is that viruses have PAMs, but the CRISPR arrays don't. So the Cas proteins use the PAM recognition as kind of a final safety check to make sure that what they're actually cutting is in fact a virus. Does it have the PAM and the spacer? Then it's a virus. Cut it. Does it have the target, but not a PAM? Then that's probably our CRISPR array. Don't cut that. Of course, viruses aren't going to go down without a fight. In response to CRISPR systems, they can evolve mutations that allow them to evade recognition by the CRISPR arrays. Or they can deploy anti-CRISPR proteins, which interfere with Cas enzymes, prevent them from doing their job. Or even deploy molecular decoys, which soak up the surveillance complexes, allowing the genome to survive long enough for them to hijack the cell. In response to that, microbes incorporate new spacers. They can deploy multiple different types of CRISPR system at once to have redundancy. And they can evolve faster Cas variants. The arms race between microbes and viruses is relentless. And it's one of the reasons why CRISPR is so diverse as a family of systems. The focus of this video is on CRISPR's role in the natural environment as an antiviral immune system. But it's not possible to really have a discussion about CRISPR without addressing its implementation as a biotech tool. In the early 2000s, when scientists were first starting to unravel the way that CRISPR works as an immune system, it was followed by a question. If CRISPR can use viral DNA to guide itself to cut specific sequences, then what if we could tell it where we wanted it to cut? By replacing the natural CRISPR RNAs with custom-designed guide RNAs, scientists were able to turn CRISPR from an antiviral immune system into a precision-targeted gene editing tool. But it's important to remember that CRISPR didn't evolve for this purpose. It evolved to kill viruses efficiently, ruthlessly, and repeatedly. And the reason that this is important is because CRISPR-Cas systems remind us that some of the most powerful technologies we have didn't come solely from human ingenuity, but also from microbes fighting for their very survival. Understanding CRISPR as a viral immune system doesn't just tell us how it works, but also why it works so well. Because for these microbes, getting it wrong meant death. So that's it for me on this episode. I hope you've enjoyed it. If you did, please leave a like and consider hitting the subscribe button and the notification bell if you'd like to see more from me. And I'll see you next time on Tiny Giants.